Hi, my name is Gabrielle Feliciano, and I interned in the Memorial Sloan Kettering Cancer Center, and we worked on mutagenesis for the enzyme SUV39H1, and we basically wanted to mutate the primers of this protein to get Y297. What is SUV39H1? It's a protein methyltransferase that adds a methyl group to a base, which is extremely important for gene silencing, regulation of the control switch for exiting the cell cycle, and entering cell differentiation. So basically what SUV39H1 is important for is differentiation, regulation, and gene silencing. The purpose of our experiment, even though I was there for maybe a week so far, but it's basically to check the expression level of SUV39H1's amino acid Y297F, the mutated form, in the presence of melanoma cells after completing the processes of Western blotting, mutagenesis, DNA sequencing, transfection, and infection. And so far I've only gotten up to the mutagenesis part, and basically this is a new approach to curing skin cancer. So we don't really have definitive results, but like the previous presentation, there's always possibilities for more results after more testing. So my mentor, Nale, she was basically responsible for most of the Western blotting process. So I didn't do most of that. I did most of the mutagenesis. And the purpose of Western blotting is it's a form of protein analysis that allows one to identify the target protein or the protein that you are looking for in the midst of a complex protein mixture, and to collect data about that protein, all through antibody-antigen interaction. And I'm going to explain that. First, the SUV39H1 protein is put through a gel electrophoresis. The position of the proteins are revealed by a sensitive protein stain. And the positions are labeled with a specific antibody that recognizes only phosphorylated proteins. And I don't really know which antibody and antigens she used because she did this project herself, but she's going to teach me more as time passes. And last of all, a second enzyme-linked antibody reveals the protein of interest, which would be Y297F. The process that I worked with most was mutagenesis, and it was a very long 3-day process because of a lot of mistakes that were made as well. So the purpose of mutagenesis is to change genetic information so that further analysis of the subsequent changes in gene expression and gene products will help elucidate the functional effects of the mutation. So we want to know what the Y297F will do in the presence of melanoma cells. The procedure is very long, but I can explain it. So basically, we aim to mutate the primers, the mutagenic primers. We aim to mutate the primer TAC to TTC within the plasmids of the SUV39H1 enzymes to test the effects of the mutation. The 2-day process, we used Quick Change Lightning site-directed mutagenesis kit, and we started by preparing a reaction with, we used a reaction buffer, distilled the H2O, 15 microliters. The plasmid DNA template, we added 0.4 microliters. We added 0.8 microliters of primer F, 0.8 microliters of primer R, 0.4 microliters of the DNTP mix, and 0.3 microliters of the quick change lightning enzyme that came with the kit. We cycled this reaction for 4 minutes and 39 seconds because that's the necessity for SUV39H1 based on its DNA sequence. And we repeatedly iced and incubated this. And then we added the DPN treatment. And because the DPN treatment is used so that the single-stranded DNA can be used instead of the template DNA. We need the mutated primers and not the template DNA. That's how we differentiate between them. And that's what we use the Western blot for, because we added, it's a really long process, and Nawe was just halfway between teaching it to me, so she basically had me do the steps and explain it later. So that was basically all the things we have done so far. And so far what we have now is the DNA with the mutagenic primers that we need, which are Y297F, and we haven't yet gotten to transforming them into colonies or purifying the plasmids or doing the transfection or the infection. So we're right in the beginning of the process. So work in progress. The DNA sequencing is going to be used to test whether or not we actually changed the TAC primer to TTC. The transfection is going to be used to make the newly mutated protein and gene into a virus, and then we're going to infect melanoma cells with this virus and test the results that it has on the melanoma cells. We haven't yet gotten to that, but I'm hopeful that we'll get pretty close to infection by the time school starts. So, special thanks to everyone here, the Harlem Children's Society, Dr. Luau, and my mentor, Ms. Nawize. Any questions? I'm giving a fair warning that we haven't done a lot yet, so I might not have your answers, but
I like how she put work in progress. I mean, that's what a lot of you expressed the same concerns, if you will. You know, I've had many conversations with many of you over the week talking about just that, the work in progress, which is fine. Some for you to explore for next week for your final written project and beyond. So, Irish?
The plasmids that you used, where did you exactly get them from?
Nawe was responsible for most of the preparation before I got there. So the plasmids, I think she got them from the kit because they were provided, like the kit provided materials for us. So we were provided with plasmids, the reaction buffer, the enzymes, and all those things. We just had to add the specific protein that we wanted, which was the SUV39H1. Yeah.
And your anticipated results, what interests you most in terms of the inspection? Any anticipated thoughts about that or results?
Well, Nawe is hoping that that the infection will show if or if not the Y297F protein will be able to stop melanoma in any way from maybe keeping it from spreading and multiplying. So that's her hope, but we're not really sure what it's going to actually result in yet. So we need to wait till we actually do the experiment.
Right, the spreading, which as we know, in cancer is the key.
That's basically the priority in most of the Sloan Kettering labs. All the people that we were working with in that lab were responsible for some type of new approach to stopping the spread of cancer. So, questions? Irish?
The infection also, you said that you guys are going to infect what you get into a virus. Do you plan on using HIV virus? Because I know that HIV was used for leukemia.
Yeah, it's been tried before, so now I'm just trying a new approach. She's not trying to, she's making, she wrote a very brief thing of what she planned to do, and she planned to make a virus, not the HIV virus, but she planned to somehow transform the SUV39H1 with the Y297F into a virus. by transfecting it with mammalian cells. So there was no mention of HIV so far as I know.
So you're not sure what virus exactly you got?
No. She knows what virus she wants to use, but I don't. Any other questions? Okay, thank you.